The cell cycle is the process by which a cell grows, replicates, and divides. It can be broken into three major stages: interphase, mitosis, and cytokinesis. Interphase is the period during which the cell is not replicating. During this time, the cell is carrying out its specialized function and growing in preparation for mitosis. It consists of the G1S and G2 phases. During the G1 phase, the cell is growing and carrying out the functions of the cell. In the G1 phase, the cell passes through its first checkpoint, at which the cell decides whether or not to divide. In the S phase, DNA is replicated or synthesized. This is the stage where the chromosomes duplicate. Each chromosome now consists of two identical sister chromatids that each have a centromere, the point where the chromatids are attached. In the G2 phase, the cell prepares to enter into mitosis. Mitosis is the period in which the chromosomes separate and the cell divides. The replication of cells is necessary for processes such as to replace damaged cells or growth. Mitosis is also used by some organisms that reproduce asexually, such as bacteria and other prokaryotes. Mitosis is followed in the cell cycle by cytokinesis, which is when the cell cytoplasm divides, creating two daughter cells. Mitosis can be broken down into four stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes begin to move toward the center of the cell and the mitotic spindle begins to form. At the end of the mitotic spindle are the centrioles. During prophase, the centrioles migrate to opposite poles in the cell. Their position determines how the chromosomes will move during mitosis. The chromosomes will eventually move towards the centrioles and the cell will divide perpendicular to the line between them. In metaphase, the mitotic spindle completely forms and the chromosomes line up along the metaphase plate, which is an imaginary line in the middle of the cell. The chromosomes are lined around the mitotic spindle, which attaches itself by spindle microtubules to each chromosome at its centromere. Note that there are two centromeres in each chromosome, accommodating for the two sister chromatids making up the chromosome in this stage. In anaphase, the centromeres of each chromosome are pulled apart and the sister chromatids become individual chromosomes. There are now twice as many chromosomes as in metaphase. The spindle microtubules pull the new chromosomes to opposite poles of the cell towards the centriole it is connected to. If a chromosome is left without a centromere, it will be left behind and randomly taken in by one of the two daughter cells. In telophase, the chromosomes are now gathered at the centrioles at the poles of the cell where the daughter nuclei begin to form. In mitosis, the daughter chromosomes are identical to the original chromosomes from which they were replicated. Thus, the daughter nuclei are identical. At the end of telophase, cytokinesis splits the original cell into two daughter cells surrounding the new nuclei. Each cell receives half the cytoplasm and organelle. Meiosis is necessary for sexual reproduction. The purpose of meiosis I is to separate homologous chromosomes. In prophase 1, homologous chromosomes pair up through synapses, forming a tetrad. The mitotic spindle begins to form and the centrioles move to opposite poles of the cell. During prophase 1, crossing over takes place where sister chromatids in the homologous pairs exchange genetic information. In metaphase 1, the tetrads line up along the metaphase plate and the mitotic spindle completely forms. The spindle microtubules are attached at the centromeres of the homologous pairs. Independent assortment of the homologous chromosomes contribute to genetic variation. In anaphase 1, the homologous chromosomes are pulled apart by the spindle microtubules, but the sister chromatids remain attached. In telophase 1 and cytokinesis, the parent cell are split into two haploid cells. However, the chromosomes are still double, so the cells must go through meiosis II. The purpose of meiosis II is to separate. In prophase 2, the nuclear membrane disappears again if it had reformed, and the chromosomes begin to move to the middle of the cell. The spindle fibers form, and the centrioles move to opposite poles. In metaphase 2, the chromosomes are at the metaphase plate and the spindle fiber completely forms. In anaphase 2, the sister chromatids separate, being pulled by the spindle microtubules and move to opposite poles of the cell. The sister chromatids are now individual chromosomes. In telophase 2 and cytokinesis, the cell splits and four haploid daughter cells result, each containing single chromosomes. There are several differences between mitosis and meiosis. Diploid cells are the result of mitosis and they have pairs of chromosomes. Diploid cells have the number 2N and are identical in genetic material to the parent cell. Haploid cells, N, are the result of meiosis. Each daughter cell has half the number of chromosomes and are genetically different from the parent cell due to crossing over an independent assortment.